This first problem is a test cross. So in a test cross, we actually have an individual who we do not know all of their genotype. So this alligator is pigmented, which means that he has one dominant allele, but we are unsure if he is homozygous dominant or heterozygous. So to fix that problem, we can then cross this alligator with a homozygous recessive individual who would show up with the phenotype albino, or no pigmentation. To solve the test cross, one of the easiest ways is to just fill in what we know in a Punnett square. So here's our Punnett square. So let's start with mom. We know that the female alligator is homozygous recessive. So her genotype is little a. We could put that on top of our Punnett square. Okay, for our male, we know he has one big A, one dominant allele, and then we have this blank over here. So we can fill in the Punnett square. We're going to have a big A, little a, big A, little a. And then in these bottom boxes, we know we're going to have a little a from mom. So we're still unsure about with dad. Now, if we go back to the question, we can see that the clutch of baby alligators from these parents included both pigmented and albino individuals. So from what we have so far in our Punnett square, we see that big A, little a, we're going to have some pigmented babies. So to get unpigmented individuals or albino alligators, we need the father to have a little a, so a recessive allele. This means that the offspring in these bottom boxes will be little a, like mom. Okay, so little a will give us albino babies. So the answer to our question here, what is the genotype of the father? We now know that he has to be a heterozygote, big A, little a, for his offspring to be both pigmented and albino. So this first problem is a case of complete dominance. Our first step is to make ourselves a key of alleles. So brown is dominant, so big B will represent our brown allele. White is recessive, so our lowercase b will be the white allele. Okay, so next thing we need to determine is our actual cross. So blank times blank. What are our parents? So an individual with homozygous dominant genotype. So that means that homozygous is two of the same alleles, and we're talking about the dominant allele. So the first parent is big B, big B. Okay, the recessive allele is the little b, so a white individual is going to have two copies of the recessive allele, and that's the only way to get that white coloration to show up. Okay, so once we have our cross, we're going to divide or segregate our gametes and put them on a Punnett square. So we'll draw our Punnett square. Okay, so first parent is big B, so we're going to put a big B here and a big B there. Second parent is little b, so we'll put those gametes on the top of our square. Once we have that, we're going to fill in our square. Okay. So one gamete from each parent. And then we have to go back to the problem and answer what is actually asked. So this problem wants to know the genotypic ratio, which is what letters are actually in our box. So the genotypic ratio is 100% big B, little b, or heterozygous. So the phenotypic ratio is going to be a translation of our genotypic ratio. Okay, so what color are these rabbits? So big B, little b, heterozygote has one dominant allele, which means that all of these rabbits are going to be brown. This second problem is very similar to the first. So we're talking about pigmentation versus albino. Okay, so a pigmented giraffe, let's use big A for pigmentation. And we'll use little a for albino. So albino is the recessive allele or a recessive trait. And so our parents, we set up our cross, we have a homozygous dominant giraffe, big A, crossed with an albino giraffe. And the only way to see the albino allele show up is to have two copies of the recessive, so little a, little a. So put those gametes on our Punnett square. First parent is big A, second parent is little a, little a. We fill in the box with one allele from each parent. Okay, then we go back and figure out what this problem is asking. So we're asking, will they yield an albino offspring? So if we look at our box, all of these are big A, little a, heterozygous, and as long as there is a copy of the dominant allele, these giraffes will have pigmentation. So there's a 0% chance of having an albino baby. So far, we have looked at a pattern of complete dominance, where one allele completely masks or hides the second allele. That's not always the case. Sometimes we see a pattern of incomplete dominance, where one allele only partially masks the other allele. So in this problem, we see that the black coat color is dominant, though incompletely dominant, to yellow coat color in labs. So when we have a heterozygote puppy, we're going to see a mix of the two colors. So black and yellow mixed gives us a brown or chocolate lab. An important part of doing incomplete dominance problems is making yourself a key. So if we have the genotype big B, we know that that is going to be a black puppy. Okay, a heterozygote is going to show up as a brown phenotype. And the recessive little b will be a yellow pup. So keep this key in mind. This will be important when we get to the last part of the question when we're answering about the phenotypic ratios. A brown lab is crossed with a chocolate lab. This is two ways of saying the same phenotype. Brown, chocolate, same thing. So we said before that brown means that we have a heterozygous individual. Okay, so big B, little b, crossed with a chocolate lab, which is also brown, so another heterozygote. And so now we've got our parents. We're going to put their gametes on a Punnett square. So there's our Punnett square. We've got big B, little b for parent number 1, big B, little b for parent number 2. Now we can fill in our square. So big B, big B, big B, little b, big B, little b, little b, little b. Okay, so the question asks for both the genotypic ratio and the phenotypic ratio. So genotypic ratio is reading the genes. So what are the actual ratios of the letters in the Punnett square? So in this case, we have 1/4 of our babies are big B, 1/2 of them are heterozygotes, big B, little b, and 1/4 of them are homozygous recessive. For phenotypic ratio, we're going to be looking at the actual colors of the dog, so what we see, the physical features. So in this case, we have to translate these big B's, little b's into an actual color. So Big B is going to be black. 1/4 of our boxes have the Big B, so we have 1/4 black puppies. Half of the boxes have Big B, Little B, so those are brown dogs. And the last 1/4 of the boxes are Little B, which means that we're going to have yellow labs.